Good afternoon. Today, Erica and I are going to be updating you on the Aplysia californica. First, let's revisit some commonly asked questions. Aplysia californica is a species of sea slug. Its diet primarily consists of red and brown algae. Ever wonder what contributed to the color of its ink? Think the toxins and pigments from its red seaweed diet. Aplysia californica reach their reproductive maturity at 85 days after hatching, and the development of their nervous system continues until they reach 140 days. Their lifespan is around 1 year. This species of sea slug is used as a model organism for exploring molecular mechanisms and neuroscience research. Most commonly, they are used to learn more about learning and memory. Characteristics that make them ideal model organisms for research includes the large size of their neurons, as well as the fact that their simple nervous systems only contain 20,000 neurons, rather than the 100 billion us humans have. The following images are dorsal and ventral views of the Aplysia californica. On the left-hand side, we have the dorsal view, and on the right-hand side, we have the ventral view. Looking at the ventral view, we can easily identify the foot, which contributes to their locomotion. The dorsal view, on the other hand, shows the parapodia, which are large flaps of skin that have been evolved to protect the organs of the sea hares. Both the head and the tail can be identified in the images. There are several reasons for which the Aplysia inks in its natural habitat. The first and most evident reason that they ink is to ward off predators. This is an effective mechanism for them, because their ink contains biological compounds that are actually toxic to other animals. The second reason for inking is to use the chemical messengers within the ink to warn other Aplysia of surrounding danger. Another reason for inking is to inhibit the growth of various microorganisms present in the environment. It is also important to mention the reasons for which they ink in the laboratory. The Aplysia ink in differing situations, including during the training protocol and when they are being moved from one surface to another. Now, we will be discussing the collection and storage of the ink. When we observe the Aplysia inking, we immediately grab a pipette and an Eppendorf tube, as seen in the image above. The ink is collected with a pipette, and we strive to attain the most concentrated ink possible. Once the ink is collected, we place it in the Eppendorf tube, which is then labeled with the Aplysia number and the date. The ink is then stored in the refrigerator for use in the near future. You now know that Aplysia ink has antimicrobial effects in the environment, and that there is a biologically active compound that plays a major role in this. This biologically active compound that was previously mentioned is escapin. Our research has been focused on demonstrating that the ink can inhibit the growth of human pathogens such as E. coli and S. aureus. We confirmed this by contrasting the antimicrobial activity of the ink to that of penicillin and tetracycline, which are both known antibiotics. As research on the Aplysia californica advances, more researchers have taken to studying the potential biomedical uses of the ink. This is why we decided to remain focused on the antimicrobial effects the ink has on human pathogens, but we went another step further. In previous years, we really had a focus on determining the ability of naturally excreted Aplysia californica ink to inhibit the growth of various bacterial species using the Kirby-Bauer test. We had also focused on comparing the antibacterial properties of the ink to other antibacterial products. This year, we have focused on obtaining consistent data confirming our findings, as well as we have begun thinking about what makes one ink have more antimicrobial activity than another. The way we did this involved a process of several steps. First, the ink would have to be collected, labeled, and stored. Then, we would use the Kirby-Bauer method, which involved disc preparation, plating on Mueller-Hinton agar, disc placement, incubation, and measuring the zone of inhibition. The final step would be the analysis of all of our data. Now, we will be showing you some of our recent findings. Here is an image of our plates that have been inoculated with E. coli and inhibited by the Aplysia ink. Our controls are simply labeled E. coli, and the antibiotic discs are labeled Tet E. coli, which stands for tetracycline. We look to the zone of inhibition, which is the area around the disc soaked in ink, which bacterial colonies do not grow. This area determines antimicrobial activity and is measured by a ruler from the center of the disc to the edge of the area with zero growth. Here, we look at discs inoculated with S. aureus and inhibited by the ink. Controls are labeled S. aureus and antibiotic discs are labeled S. aureus pen, with pen standing for penicillin. Further trials are shown above. This particular trial was testing against S. aureus. Here we observe another trial against E. coli.
For our first round of trials with our E. coli, on 2 of 20, the tetracycline had 14 millimeters zone of inhibition. 3 of our Aplysia did show that they do carry some antimicrobial properties, E, C, and A. Our C. Aplysia had the most with 5 millimeters of zone of inhibition. Our E. Aplysia had 4 millimeters zone of inhibition, and our A. Aplysia had 1 millimeter zone of inhibition. B1, B2, and B3 Aplysia all showed no antimicrobial growth on this trial, and our F. Aplysia, unfortunately, the results were inconclusive, which is why we put an asterisk there. In this graph, tetracycline has the greatest zone of inhibition as it did with the previous graph. A, C, and E Aplysia Inc. are staying consistent with the greatest zone of inhibition, which means that they have the greatest antimicrobial properties. On this trial, B2 and B3 are showing minimal antimicrobial properties that they did not show previously. They both show a zone of inhibition of 1 millimeter. Unfortunately, again, the results for F were inconclusive. With this trial, penicillin only showed 0.5 millimeters of a zone of inhibition. For this trial, penicillin had the greatest zone of inhibition with 18 millimeters. Aplysia Inc. C was very close to penicillin with antimicrobial properties, showing a 13 millimeter zone of inhibition. Aplysia A's Inc. also showed great antimicrobial properties with 9 millimeter zone of inhibition. Aplysia B2 and F showed minimal antimicrobial growth, with B2 having a zone of inhibition of 1.5 millimeters and F having a zone of inhibition of 0.5 millimeters. Unfortunately, the results for B1, B3, E, and tetracycline were inconclusive. In this second trial of Staph aureus done on 2 of 20, penicillin has the greatest zone of inhibition of 17 millimeters. Aplysia C and Aplysia A are staying consistent with having the greatest antimicrobial properties close to penicillin, with Aplysia C having a zone of inhibition of 14 millimeters and Aplysia A having a zone of inhibition of 9 millimeters. In this trial, we were able to get a good look at the antimicrobial properties of Aplysia E. This Aplysia shows great antimicrobial growth along with A and C, and penicillin with having a 11 millimeter zone of inhibition. F and B2 still showed minimal microbial properties, with F having 0.5 millimeters, which is consistent with the last trial, and B2 shows 1 millimeter zone of inhibition, as it showed 1.5 millimeters on the last trial. B3 does not seem to show any antimicrobial properties in this study. Again, the results for tetracycline and B1 were inconclusive. In this trial, tetracycline shows the greatest zone of inhibition with 13 millimeters. Aplysia Inc. E has the greatest antimicrobial properties than the other inks with a 3 millimeter zone of inhibition. Aplysia Inc. C shows 2 millimeter zone of inhibition, and Aplysia B2 and F both showed minimal antimicrobial properties with 1 millimeter zone of inhibition. These Aplysia inks do show more antimicrobial properties than the common penicillin, which has a zone of inhibition of 0.5 millimeters, along with our Aplysia Inc. A. Our Aplysia Inc. of B1 does not show any antimicrobial properties.
In E. coli Trial 2, which was done on February 26th, the results of inks A, B1, C, and F all improved. Tetracycline remained consistent with an 11 millimeter zone of inhibition, while ink C approached similar results with a 6 millimeter zone of inhibition. E also remained consistent in its results and had a zone of inhibition of 3 millimeters, as it did in the previous trial. Finally, penicillin had consistent results as well and remained at a 0.5 millimeter zone of inhibition.
In this trial, tetracycline shows the greatest zone of inhibition of 9 millimeters. Aplysia C comes up very close behind in antimicrobial properties with a zone of inhibition of 8 millimeters. Aplysia E and A also showed great antimicrobial properties, with E showing a zone of inhibition of 6 millimeters and A showing a zone of inhibition of 4 millimeters. Penicillin only showed a zone of inhibition of 0.5 millimeters, which was the same as Aplysia B1 and Aplysia B2. This data shows that the ink of Aplysia A, C, and E were all more successful than the common antibacterial medicine penicillin. The results for F were inconclusive. For this trial, penicillin showed the greatest zone of inhibition with 16 millimeters. This is completely different from the last trial, which only showed penicillin with a 1.5 millimeters zone of inhibition. Comparable with trial 1, Aplysia C has the greatest antimicrobial properties with a zone of inhibition of 12. Also, Aplysia E and A showed great antimicrobial properties, with E having a zone of inhibition of 8 millimeters and A having a zone of inhibition of 3 millimeters. On this trial, tetracycline showed a lower zone of inhibition of 8 millimeters. Still, the ink of C, E, and A showed great antimicrobial properties. Unfortunately, the results for Aplysia ink B1, B2, and F were inconclusive.
Throughout our research, we found the following to be conclusive. The three inks that were most consistent in producing the most effective antimicrobial results were inks A, C, and D. Amongst these three, C had the highest results for 6 of the 8 trials. Inks B3 and F showed less effectiveness in their results, although some of the results were inconclusive due to sliding of the disc. B1 and B2 were similar in that their ink showed little to no antimicrobial properties, with some inconclusive results as well. Data for all inks remained fairly consistent in that A, C, and E had the greatest zone of inhibition, and B1, B2, B3, and F had the lowest. In many of the trials, there were similar results between the antibiotics and inks C and E. For instance, in Staph aureus Trial 1 on February 26th. In this trial, the zone of inhibition for tetracycline was 9 millimeters, while that of ink C was 8 millimeters and that of ink E was 6 millimeters. Another interesting result was that of Staph aureus Trial 2 on February 26th as well. In this trial, ink E and tetracycline both had a zone of inhibition of 8 millimeters. Meanwhile, penicillin had a zone of inhibition of 16 millimeters and ink C of 12 millimeters. Our results illustrate the antimicrobial properties of Aplysia ink and raises the question of why some inks show more effectiveness than others. This semester, we began investigating the differences between the inks. Therefore, we introduced the gel electrophoresis technique in our lab so that we can separate charged molecules according to size. As you can see in the image above, the individual ink samples move through the gel differently from one another. Unfortunately, the results were not definitive, but we hope to further investigate this and obtain more data. The way the DNA bands move is according to charge and size. Speaking of charge, negatively charged DNA will move away from the cathode, which is negative in charge, and move towards the anode, which is positive in charge. In regards to size, smaller molecules can move faster and more easily through the gel, while larger molecules and fragments move slower. Because of the pandemic, we had to put this work on hold, but we will pick right back up where we left off. We are so excited to announce all of our upcoming research. From our data, we can see that ink C and E seem to consistently be top contenders for the most antimicrobial activity. Meanwhile, inks B2 and B1 seem to be the least effective. Doesn't it make you wonder, what makes one more effective than the other? Is it the dyed of the Aplysia? Is it the consistency of the ink? We will be exploring this in upcoming semesters. We will additionally be performing more gel electrophoresis runs in order to separate the DNA fragments according to size and charge. This will help us further answer the questions we have. We had done a few gel runs throughout the semester, but unfortunately the pandemic had stopped us from finding any definitive evidence. We also hope to add in some thin layer chromatography in order to identify compounds and determine their purity. Finally, we hope to incorporate Western blot testing to detect certain proteins. Research with Dr. Hale was an amazing experience that has furthered all of our knowledge on the topics of neurobiology, molecular biology, and microbiology. It has taught us many things about working in a team research environment, including patience, determination, creativity, and teamwork. Because it is our senior year, we will no longer be able to physically be there, but we will always be excited to hear about new findings and will always be willing to support the team. We are so thankful to have been given the opportunity, and it is all because of Dr. Hale.